In this video, we'll start to explore the structure of nucleic acids, starting with the basic building blocks. The monomer of nucleic acids is the nucleotide. Nucleic acids are polynucleotides. And all nucleotides have three parts: one or more phosphate groups, a pentose sugar, which means a sugar with 5 carbons, and a nitrogenous base. Let's look at each of these three elements. First, the phosphate group. The phosphate group might look like this, or it might look like this, or even this. Same thing. Remember that oxygen is extremely electronegative, and sometimes the hydrogen atom will simply leave, but oxygen keeps the shared electrons. So hydrogen leaves as a hydrogen ion, basically a single proton. This gives the phosphate group 1 or 2 negative charges. By the way, let's review. What do we call something that increases the concentration of hydrogen ions in solution? Hopefully you recall that the word we use is acid. And by the way, this is why we call DNA and RNA nucleic acids. The nucleic part, by the way, is because they were first described as being located in the nucleus. And of course, we have a lot of DNA in the nucleus of eukaryotic cells, but RNA isn't necessarily localized to the nucleus. We find some of it in the cytoplasm as well, and DNA isn't constrained to the nucleus either. Even eukaryotes have DNA outside the nucleus, in the mitochondria and chloroplasts of cells. And of course, prokaryotes don't even have a nucleus, so their DNA and RNA is just hanging out in the main compartment of the cell. Let's move on now to the pentose sugar of a nucleotide. We have 2 possible pentose sugars, ribose and deoxyribose. Ribose is found in RNA and deoxyribose is found in DNA. And these sugars, of course, give them their names, ribonucleic acid and deoxyribonucleic acid. Let's compare the structures of these pentose sugars. They are identical except for one little detail. Here on the 2' sugar, ribose has a hydroxyl group, but deoxyribose does not. It simply has a hydrogen instead, and this is reflected in the name. Deoxyribose, meaning ribose, deoxy, minus an oxy, or oxygen. Let's count the carbons now because this is important. We always number them starting here. This is the 1' carbon, the 2', 3', 4', and 5'. The phosphate group is attached to the 5' carbon, which is outside the ring, and the 3' carbon has a hydroxyl group in both ribose and deoxyribose. The 3' carbon is where we're going to attach another nucleotide when we start putting nucleotides together to make a polynucleotide. When we do that, we make covalent bonds between nucleotides. When we make a polynucleotide, we see an alternation of phosphate group, sugar, phosphate group, sugar. So we sometimes call this the phosphate sugar backbone of a nucleic acid. The bases are not involved in making covalent bonds to other nucleotides, but of course they're super important. The 1' carbon is where the nitrogenous base is attached. And here we have several choices. If we're looking at a nucleotide of DNA, this nitrogenous base could be adenine, guanine, cytosine, or thymine. And if we're looking at a nucleotide of RNA, the nitrogenous base could be adenine, guanine, cytosine, or uracil. We generally refer to these bases by their first letter, A, C, G, T, or ACGU. These nitrogenous bases can have 2 interlocking carbon-nitrogen rings, in which case we call them purines, or they can have a single carbon-nitrogen ring, in which case we call them pyrimidines. These nitrogenous bases can form hydrogen bonds with other bases, and base pairing is specific. If we're looking at a molecule of DNA, we have 2 polynucleotides, or strands, that are bound together by hydrogen bonds between the bases. Notice that we have 2 strands here of DNA, and also notice that they're running in the opposite direction. This one is going from 5' to 3', and the other one is going 3' to 5'. This is called an anti-parallel orientation, and it is necessary for base pairing. We're not going to see 2 parallel strands running in the same direction and base pairing with each other. Not going to happen. So, in a molecule of DNA, adenine can pair with thymine, and cytosine can pair with guanine, and that's it. If you need a handy mnemonic to help you remember this, think apple tree, AT, chewing gum, CG. This pairing is accomplished via hydrogen bonds, shown by the dotted lines. 3 hydrogen bonds between C and G, and 2 hydrogen bonds between A and T. If we have a strand of DNA and a strand of RNA. And this is going to happen, by the way, during transcription when information in DNA is copied to RNA. The same base pair rules apply, except of course RNA doesn't have T, it has U instead. So A pairs with U, C pairs with G, G pairs with C, and the T in DNA will pair with an A in RNA. What about RNA? Well, RNA is usually single-stranded. Now, there's no biochemical reason that it can't be double-stranded. It totally can. In fact, we see double-stranded RNA in many viruses, where it acts as their genome. For example, rotaviruses, which cause diarrhea. They're the most common cause of diarrheal disease in children. In fact, just about every human on Earth has been infected by a rotavirus at least once by the time they are 5 years old. And we as eukaryotes have developed defenses against these viruses, including cutting up double-stranded RNA in our cells. This is accomplished by an enzyme called dicer, among other proteins, and it slices and dices double-stranded RNA into tiny fragments. So RNA in our cells is generally single-stranded, but it can fold on itself and make interesting structures, again using base pairing. For example, transfer RNA folds to make this interesting cloverleaf structure, which is based on hydrogen bonding between G, C, and hydrogen bonding between A and U. And then it twists into a tertiary structure. Okay, hopefully this video helped you understand the basics of nucleotide and nucleic acid structure.